transferred to the coenzyme for further, you can say, conversion, or you can say for the further collection in the ETS, that is the electron transport system. So if you think about it, in the aerobic respiration, the TCA and the this thing, electron transport system, they are sort of connected, very connected with each other. They go hand in hand. Now, talking about this particular cycle, the cycle is also called as citric acid cycle, because we know that the first formed product is citric acid. Or tricarboxylic acid cycle, because all of the compounds, they have 3 carboxyl groups in them, right? We saw that. All right. Now see, what actually happens in this particular cycle? So here we saw how in the case of oxidative decarboxylation, the pyruvic acid, it gets converted into acetyl coenzyme A by the pyruvate dehydrogenase complex. Now, this acetyl coenzyme A, it sort of condenses with the oxaloacetic acid. So it starts from here. So the acetyl coenzyme A condenses with the oxaloacetic acid and forms the first product, that is the citric acid. The enzyme that causes this condensation reaction is called as citrate synthetase. Now, from that citric acid, this aconitic acid is formed. Okay, so now the citric acid gets converted into aconitic acid by the action of the enzyme aconitase. Then this aconitic acid gets converted into isocitrate. Sort of, this is like a isomerization reaction wherein it changes into a different form called as isocitrate by the action of the enzyme aconitase. Then the isocitrate gets converted into oxalosuccinic acid by the action of the enzyme isocitrate dehydrogenase, which happens in the presence of manganese. After that, the oxalosuccinic acid, they get converted into alpha ketoglutaric acid, and then the alpha ketoglutaric acid gets converted into the succinyl coenzyme A. Now, during this step, the NADH2 molecule is being synthesized and the CO2 molecule is being released. And this coenzyme A is also being added. From the succinyl coenzyme A, it gets converted into succinic acid. And from the succinic acid, we get the fumaric acid, during which FADH2 is synthesized. Just like how there is NADH2, same way, another version of NADH2, we have the FADH2. Then the fumaric acid gets converted into malic acid, and lastly, the malic acid regenerates the oxaloacetic acid so that the cycle can keep on going uninterrupted. So this is called as your Krebs cycle. See, they won't ask you to make the steps of this. You should know what is happening, but they'll not ask you to create this in the answer. So all in all, but this reaction is very important, that pyruvic acid along with 4 molecules of NAD plus FAD plus along with 2H2O, ADP and inorganic phosphate in the mitochondrial matrix, 3 molecules of CO2, 4 NADH2, FADH2 and ATP. This is the end product of the Krebs cycle. Clear, students?
Yes, ma'am.
Then we have the electron transport system, the ETS. Now see, in all of these areas, we saw either FADH2 was being synthesized or NADH2 and all of that. Now let us see how is it used and where is it used in fact. So when we talk about electron transport system, the ETS. Now here, the electrons, wherever the electrons are gained, they are passed on from one carrier to another carrier, and this whole entire system is called as the electron transport system. Okay, so the electron gets transported from one carrier to another carrier. Clear? Where does it take place? So it takes place, always remember the location. So it takes place in the inner mitochondrial membrane, towards the inner membrane of the mitochondria. Now let us see, I told you that the electrons, they are passed on from one carrier to another. So complexes are needed for that. Okay, so there are 4 complexes that are needed for the electron passing. First complex is NADH dehydrogenase. Second complex is succinate dehydrogenase and FADH2. Third complex is, these are the names of the complex. Okay, nothing complicated in it. Cytochrome BC1 is the third name. And then cytochrome C oxidase is the fourth complex. So all in all, these 4 complexes are needed during the ETS or the electron transport system. Now let us see what happens during the ETS. Firstly, now NADH, which is produced in the mitochondrial matrix. We know that during, we just saw in the citric acid cycle, NADH2 was being made, right? So now that NADH2, it gives us the electrons, right? And the electrons from that NADH during the citric acid cycle, they are oxidized by NADH dehydrogenase, which is the complex number 1. So these electrons, they are received by the complex 1. From the complex 1 or from the NADH, the electrons are then transferred to ubiquinone. Now, ubiquinone is a mobile carrier. Mobile means it can move. Okay. Now, what happens? Ubiquinone receives reducing equivalence via FADH2, because we know during the TCA cycle, FADH2 is also being synthesized. So that also gives the electrons, and the ubiquinone receives from both of them. Okay, so basically here, after that, once the ubiquinone receives the electron, it gets reduced, right? So it becomes ubiquinol. So from ubiquinol, it becomes ubiquinol. Okay, and then with the transfer of electron to cytochrome C via cytochrome BC1 complex. Now, see, cytochrome BC1 complex, it is the complex number 3. So now the electrons from the ubiquinone, they are transferred to the cytochrome C complex through cytochrome BC1. Also, talking about the cytochrome C, so it is a small protein attached to the outer surface of the inner mitochondrial membrane, and they act like a mobile carrier. Sort of, it is like, imagine it like a toy car moving between complex 3 and the last complex. So it takes the takes the electrons and moves to and fro between these 2 complexes. At the end, once this whole process is done, oxygen is the final electron acceptor. Many a times they ask this, which molecule is the final acceptor of the electron? So oxygen. Now, after it receives the electron, it becomes reactive and then it forms the water molecule. And that is how the ETS gets completed. Clear, students? These are all the steps. Nothing you can say different other than this happens. It's just that you people should know that how the electrons are taken from the energy intermediates that are formed during the citric acid cycle, and then it is transferred from one complex to another. Ultimately, at the end, it gets received by the oxygen, which is the final electron acceptor, and then that forms the water molecule. Clear? Any doubts?
No, ma'am.
All right. Now, the next thing is oxidative phosphorylation. Now, see, oxidative phosphorylation, you people remember I told you that phosphorylation in general means basically referring to the formation of ATP molecule, isn't it? Because phosphorylation is addition of phosphate. So we know that when ADP gets added with the inorganic phosphate, it forms the ATP molecule. So now we are going to see how does the process of formation of ATP takes place. Now, when the electrons are passed from one carrier to another carrier via complex 1 up until the 4th complex, In the ETS or the electron transport chain, they are coupled to ATP synthase. Now there is another complex called as the complex number 5, which is called as ATP synthase. This complex is important for the formation of ATP. Okay, how is ATP formed? Whenever the ADP, that is adenosine diphosphate, whenever that combines with inorganic phosphate, the ATP molecule is formed, right? This is how it happens. Now, for the formation of ATP, there has to be proton, there has to be a certain process through which it happens. So that is proton gradient. Gradient you people understand, right? At one place, proton will be high and at one place it will be very low. So high to low condition is a gradient. The gradient of proton. So obviously we call it as the proton gradient. This can also be called as proton motive force. So this is required for phosphorylation. And how is it obtained? Where does this proton come from? So electron transport system. That is why we call it like, because during the ETS, oxidation reduction reactions are taking place. So therefore, this whole entire phosphorylation becomes oxidative phosphorylation. Because the protons, they are ultimately coming as a result of oxidation reduction. Now, the mechanism of membrane-linked ATP synthesis is explained by chemiosmotic hypothesis. Now, there is this hypothesis which is given by Peter Mitchell or T. Mitchell. Now, he told us that this is the possible way through which the ATP synthesis takes place. Now, see what happens. Inner mitochondrial membrane is permeable to protons, meaning it allows proton, but only in the F0 or F1 region. Now see students, do you see this whole entire part? This part? This is the structure of ATP synthase in complex 5. Okay. So the F0, F1, these are the two components of it. So these are proton permeable. Okay, they permit the proton to come through them. Energy released during the electron transport system is utilized in synthesizing ATP with the help of ATP synthase, which is complex number 5. This complex contains two major components, F1 and F0 or F0. Now, the F1 headpiece is a peripheral membrane, meaning it is at the periphery, whereas F0 is integral towards the inner side. Clear? Now, the F1 contains the site for the synthesis of ATP from ADP and inorganic phosphate, whereas the F0 or F0, which is towards the inner side, okay, it is the integral membrane protein. So it forms the channel. Channel means the pathway through which the protons would travel. The passage of protons through the channel is coupled to catalytic site of F1 component. So whenever the protons pass through this ATP synthase, then ATP molecules are produced or synthesized. So always remember, for one ATP, two protons or two H plus ions have to pass through the F0 from the intermembranous space to the matrix down the proton gradient. Down the gradient means from high gradient to low. Meaning from high concentration to low concentration, the protons have to flow, because of which or as a result of which ATP will be synthesized. 3 pairs of electrons are pushed during oxidation of each NADH. And remember, see, NADH2 or FADH2, they are also responsible for synthesizing ATP. So one molecule of NADH, it can synthesize 3 molecules of ATP, whereas one molecule of FADH2 can synthesize 2 molecules of ATP. Always remember this. Okay. So if 2 molecules of NADPH are there, so 6 molecules of ATP are there. Okay. Just like that, they can ask you to calculate. Like, for example, in a given reaction, there are 4 molecules of NADH synthesized. So how many molecules of ATP will it give us?
12 molecules.
Exactly. Because one of them yields 3 ATP. So 4 of them will yield 12 ATP. Okay. So this was oxidative phosphorylation and the ETS. Now tell me students if you have any doubts. Okay. All right, students. Now you can note it down. See, I think you have the upper part written. This part is there because it was there in the notes. You just have to note down the cycle, the names of the enzyme. Okay. So now you can note it down.
Done, ma'am.
All of you are done, students. Students, are you done? Yes or no?
Done, ma'am. Done, ma'am.
All of you are done, students?
Yes, ma'am.
Done, ma'am. Done, ma'am. Done, ma'am. Done, ma'am.
All of you are done, students.
Yes, ma'am.
All right. Okay. Okay. So now we are going to see the respiratory balance sheet. So up until now, we saw glycolysis, we saw TCA, which is citric acid cycle, we saw ETS, right? So in all of these reactions or processes, ATP was being synthesized. So just like how balance sheet is basically doing like the calculation of stuff. So here we do the calculation of how many ATP we have received. So during glycolysis, directly we got 2 molecules of ATP and we also got 2 molecules of NADH2. So 1 molecules of NADH2 makes 3 ATP, so 2 would make 6. Then in the link reaction, meaning from when the pyruvic acid was converted into acetyl coenzyme A, so during that we received 2 NADH, that means 6 molecules of ATP yet again. And in the TCA cycle, we had 6 molecules of NADH2, then 2 molecules of FADH2. So 18 molecules of ATP from 6 NADH, 4 molecules because 1 FADH makes 2 ATP. So 2 makes 4 and directly we get 2. So all in all, at the end, 38 ATP molecules are there. Always remember that in most of the eukaryotes, the net gain of ATP is 36 molecules during aerobic respiration. This is like 38, like 36 to 38 is the amount of ATP that we get. Clear, students? This is clear?
Yes, ma'am.
This is like basically whatever we have studied up until now, wherever there is like ATP molecules synthesized or formed, we are just jotting it down or noting it down. Now, after that comes the amphibolic pathway. Now, see, we know amphibolic. See, you people know what catabolism and anabolism is? Do you know, students? Catabolic, anabolic. No? See, catabolic reactions or stuff, it's when the molecules are, you can say, broken down so as to release energy. They are broken down. And anabolic is when they are synthesized. Amphibolic is in between them. Okay, it's like a dual thing. See, we know that for respiration, the best respiratory substrate is the glucose. And all carbohydrates are usually converted into glucose before they are used for respiration because glucose is the favored source or favored substrate for respiration process. Other substrates can be fat molecules or protein molecules, but they are also converted into glucose and then only they enter the respiratory pathway. So the fat, they are broken down into glycerol and fatty acid, and then the fatty acid degrades into acetyl coenzyme A and then enter the respiratory pathway. The glycerol gets converted into phosphoglycerolaldehyde and that is what happens to it. So remember that the fat and proteins, they do not directly undergo the process of respiration. Rather, they get converted into certain intermediates and then they undergo the respiratory pathway. Talking about proteins, so proteins are the ones that they are converted into amino acid and then they get the entry towards the respiratory pathway. Now see, like I told you, metabolism is anabolism plus catabolism, meaning it has molecules are synthesized in it and energy is utilized at that time. Just like how in ATP synthesis also, energy is utilized. Whereas in catabolism, molecules are broken down and energy is released. So these are the two things. But if we talk about the respiratory pathway, it is not anabolic, it is not catabolic. Rather, it is amphibolic. It has a dual thing, meaning it is anabolic along with catabolic, both of them. Clear, students?
Yes, ma'am.
If someone asks you what is the nature of the respiration or the respiratory pathway, so you see that many places there are ATP molecules and different kinds of molecules that are being synthesized, and in many places they are being utilized. So depending upon that, respiratory pathway is amphibolic. Amphi means dual. Coming on to the last topic of this chapter, that is respiratory quotient. So basically respiratory quotient is how much of carbon dioxide is released when the oxygen is consumed. So that is the respiratory quotient. Like during the respiration cycle or pathway, how much oxygen is being consumed, and as a result of which how much CO2 is being released. That is what we call as the respiratory quotient. Now talking about the respiratory quotient, it will vary from substrate to substrate. Okay, like we know that carbohydrates, fat and protein, these are the three substrates that can undergo respiration. So accordingly, the respiratory quotient would vary. For example, if we talk about the carbohydrates. So here, if you look at this reaction, that C6H12O6, which is glucose, when it reacts with 6 molecules of oxygen, so it gives out 6 molecules of carbon dioxide. So the number or the amount of oxygen that is utilized and the carbon dioxide that is given, it's the same. So respiratory quotient for carbohydrates comes out to be 1. For the fat molecule, if you see here, the respiratory quotient will be less than 1. It will be 0.7, less than 1. Okay, for fat, always it will be less than 1. For carbohydrate, it will be 1. And for protein, it will always be, like for protein, it will be 0.9. For oxalic acid, it is 4. For malic acid, it is 1.33. Okay. And when we talk about the respiratory quotient for anaerobic respiration, because we know in anaerobic respiration, there is no oxygen. So anything that is divided by 0 is infinite, right? So the respiratory quotient for anaerobic respiration is infinite. But for carbohydrate, it's 1. For fats, it's less than 1. For protein, it is 0.9. And then what are the two things? Oxalic acid, it's 4. And malic acid is 1.33. Okay, respiratory quotient, what is it? It is the amount of oxygen utilized and the ratio of how much carbon dioxide is released. So it's the calculation of that. Lastly, we have the differences. This is just an extra thing that I've written. Fermentation and aerobic respiration, the differences in between them. Fermentation is partial degradation, right? Partial breakdown happens. In aerobic respiration, complete breakdown happens. The net gain of ATP is 2 in the case of fermentation, whereas 36 to 38 ATPs are gained in the case of aerobic respiration. Oxygen is not required in the anaerobic case, and oxygen is required obviously in the aerobic. End products of fermentation are lactic acid or alcohol. End products of aerobic respiration are CO2 and water. Clear, students?
Yes, ma'am. Clear.
Any doubts up until now? So with this, we have actually completed the chapter. So now you people take down the notes.
Done, ma'am. Done, ma'am.